Who's that? One that's all this liquid tissue, but it doesn't have this stuff around the outside, or it doesn't have circles or anything.
All right. Hello, everyone. Good afternoon. I know that you guys are stressed with assignments, including mine. You shouldn't be stressed with mine because you remember my due dates are guidelines only. I know all of you will automatically want a 48-hour extension, so let's just give you a 7-day extension. All right, next Friday, if you want. All right, so please don't stress and please just do a good job. If you want yours back, you can have it back.
You're making a relation salary, thank you.
No, but this goes for everything that I want you to do, because I would prefer that you spend some time and do a good job than rush to meet a deadline that, you know, just piles up on you for whatever reason. And we all have reasons in our lives, including other units. So just chill and do a good job. Hand it in to me, hopefully next week. And if not, send me an email to say, actually, I need a bit longer. Week 13, Friday, is actually a due date for everything, because I have to be given enough time to mark everything to actually result it to the university. So we have this deal, right? And the deal from your end is to not tell the university. Oh shoot, I'm recording again. Bloody hell. All right. Don't worry, they don't listen to this stuff. Do not use this against me. All right. My very great pleasure to introduce someone who is familiar to a lot of you, particularly those of you who do biochemistry, and that is Sonia. So what you probably don't realise is Sonia actually has a very well-functioning, well-populated laboratory full of scientists over at Tribe, and she does some really cool stuff using small peptides. And those peptides, the little creepy crawly in the bottom corner, her original peptide came from a spider. So she's going to tell you all about the work that she does with these peptides and how she's designing them for novel cancer therapeutics. But you probably actually know enough about Sonia, and if not, she can tell you. But I just don't want to get in your way. So
Thanks, Sally.
All yours.
Thanks for having me, and great to see some familiar faces. So what I'm going to tell to you about today is some novel cancer targets and therapeutics. And as Sally mentioned, I'm going to talk a little bit about my research on peptides. Before we get into that, just a reminder, how can we treat cancer? So there's all these options, and there's probably more that are coming now. But the ones we are interested as scientists, or the ones that we can work on, are on molecular approaches, so molecules that can be developed to treat cancer. So there's a lot of success with hormone therapy for breast and prostate cancer, so that's been saving many lives. Immunotherapy has been a major lifesaver for some types of cancer. So this type of therapy works by having antibodies that activate the immune system. So instead of killing tumor cells directly, they just make our immune system kill them. There's target therapies that relate to therapeutics, so molecules that can specifically target a unique protein or oncoprotein that is overexpressed in tumor cells. Chemotherapy, which is the traditional way of treating cancer, that uses therapeutics that can kill rapidly dividing cells. Normally those ones target DNA in those rapidly dividing cells like tumor cells, but they're also going to target, unfortunately, other types of cells that are also rapidly dividing, and this includes hair follicle, blood cells, and digestive tract, and that's why there's so many side effects with chemotherapy, which I'm sure you are all aware. And emerging field is the radiopharmaceutical. So radiation therapy has been used for a long time now, but now it's emerging as a targeted therapy in which the ionizing radiation is attached to a peptide or a ligand that can specifically recognize a receptor on the surface of tumors. So this is now also emerging as a really niche molecular approach, and there's been some success, and there's some target radiotherapy now available to treat some types of prostate cancer. So how can we, so although there's all these strategies, there's still a lot we can do to improve cancer treatment, because there's still a lot of challenges in cancer treatment. So how can we treat new cancers in different ways, or treat all cancers with new strategies? So we need to first understand and characterize novel and specific cancer targets for a unique cancer. And I guess that's why you are here doing cancer biology. So it's very important to understand cancer biology. And you probably have seen this figure already and understand the different hallmarks and all the molecular pathways that are involved in cancer development. So these will help design therapies that are more specific towards cancer cells, that are going to be less toxic to healthy organs or cells. And this way we can develop therapeutics that are going to target a specific cancer. So we're going to have drugs that are developed to treat skin cancer and others that are developed to treat unique or specific breast cancers. But how can we develop targeted therapies that modulate these molecular mechanisms that are involved in cancer? So the first thing we need to consider is, as any real estate agent is going to tell you, location, location, location is very important. And that's because depending where our target that we want to modulate is located, we need to think what kind of therapy can we develop for that target. So if our target is in the surface of tumor cells, that's normally an easy one to target because we just need to get something to the surface of the cell. So normally if it's a cell surface target, one strategy that can be used is the use of antibodies or proteins because they can achieve very high specificity, and so we can easily modulate those targets. So a very important example is, for instance, Herceptin, which is an antibody that has been developed to treat certain types of breast cancer that express a HER2 receptor on the surface of those tumor cells. So, and what that antibody does, it inhibits an interaction in a membrane receptor, and by inhibiting that interaction, then decreases the proliferative capacity of those cells. But if you want to target a protein or a molecule that is located in a different location in the cell, like the cytosol or inside an organelle, then we need to think what kind of molecules can we use to target intercell targets. So when we have intercell targets, that means they need to cross a cell membrane. And to cross cell membranes, normally the type of molecule that is used is what is referred to as a small molecule drug. So small molecule drugs are molecules that normally are small, as the name suggests. They have about 500,000. This is the kind of things that we can take orally, like if you take an aspirin or any of those pills that we take at home, even just if it's a painkiller. This is an oral. All these oral molecules, they are small. Normally, they are based on small molecule drugs. Antibodies, proteins, they normally are injected because they are not going to survive. They are not orally bioavailable, so they're going to be degraded through our digestive system. So small molecule drugs have been very popular to target intercellular proteins. And what they do is that they can interfere with the protein of interest, and then they change or modulate an intercell pathway that could develop and induce the cancer. The second very important consideration when developing target therapies is to look into the target that we are trying to target. So the location of the target is an important one, and then look into the properties of our target. So does this target has a unique structure? Does it has unique properties? So the first thing when we are using small molecule drugs to inhibit or modulate intercell targets, we need to look, can a small molecule drug interact with our target in a way that is going to modulate its activity? So a very common type of proteins that are targeted by small molecule drugs are kinases, and that's because kinases, they have pockets that can be easily modulated by small molecule drugs. So the example shown here on this figure is the ABL kinase, which is a kinase that is expressed in chronic myeloid leukemia. And this one, because it's a kinase, that it has an ATP binding pocket where ATP inserts for the kinase to be active. And so these type of pockets are very easy to target by small molecule drugs because small molecule drugs can be developed as mimetics of ATP, and so they get inside the pocket and make the enzyme inactive. So that makes this type of enzyme or this type of targets very easy to modulate. And then the other very important thing that we need to consider is this protein or enzyme kinase unique to cancer cells? Because you don't want to inhibit kinase that is important for function in the healthy cells. So the best targets are the ones that are mutated or only expressed in cancer cells, because then you just want to target or you just want to specifically target or inhibit these proteins in cancer cells. So as I mentioned, ABL kinase is a very good example, and is one of the kinase that has been used to develop targeted therapies. So ABL kinase is an enzyme that is expressed in chronic myeloid leukemia, and this enzyme in healthy cells exists in the nucleus. So in healthy cells, it's located in the nucleus, but patients that have chronic myeloid leukemia, they have this fusion gene where the BCR, with another protein, is fused with ABL. And as a consequence, the ABL kinase, instead of being located in the nucleus, is going to be located in the cytoplasm. So the cells that are the tumors that have this BCR ABL expression, they have this enzyme located in the cytoplasm, which is in contrast with healthy cells that have the ABL kinase in the nucleus. So this was a model, is often referred as the model cancer to develop target therapies, because there's a distinct location and a distinct kinase when it's in healthy cells compared to cancer cells. Yes.
Sorry, just back to like fusion gene. So it's not a mutation in that gene; it's fused to another working gene. Or?
Yeah. So in this case, there's two chromosomes that have like a fusion. So there's like a translocation on the chromosomes, and as a consequence, this protein or these two genes fuse in the new when one of the arms of the chromosome fuse with the other.
Oh, right.
And then so it forms this new gene that express this longer protein.
Okay, so it's parts of two different genes. Okay, cool.
What about Philadelphia?
Yeah, it's called the Philadelphia chromosome that has this mutation. So what happened then is this BCR ABL kinase, once it's in the cytoplasm, is constitutively active, which means it's not regulated, and so it's going to phosphorylate its substrate in an unregulated way, so it's going to be activating all these pathways. And as a consequence, it's going to increase migration of these cancer cells, it's going to increase differentiation, transcription, and it's going to switch off apoptosis. So these cells have an unregulated proliferation. So we have now cancer cells that are proliferating in an unregulated way because of this enzyme. So what has been done to try to target this kinase was to look at the full protein. So the protein, there's the BCR part and the ABL part. And it was identified that if we could inhibit the ABL part, that was enough to switch off the activity of this fused protein and to switch off the cancer. So the strategy used was, so this is just a representation. So this is the kinase part of the ABL. So this ABL part has an ATP binding pocket and a catalytic site. So the ATP binding pocket, it's an anomaly easy one to target. So that's what it has been done. So there's the first molecule developed to inhibit this kinase is called imatinib, and that's the structure here. So this molecule kind of mimic the ATP overall structure and can easily can fit very specifically in the pocket of this kinase. So this the advantage of these molecules can target the BCR-ABL in the cytoplasm of cancer cells, but it cannot target ABL in the nucleus of healthy cells. So imatinib stays in the cytoplasm, it doesn't get into the nucleus, so it's only going to inhibit the function of the mutated BCR-ABL kinase. So this molecule is actually, yes.
Oh, sorry. What does the BCR part of this protein actually do other than move the protein to the cytoplasm?
So it has a biological function, which I don't know what it is, but when it's fused with this, it just makes it move into the cytoplasm and be constitutively active.
BCR stands for breakpoint cluster region, and that doesn't tell you anything about what it does. But it's constitutively expressed, so we would probably say it's like a housekeeping. Yeah, it's boring.
Yeah.
It's boring. It did something.
Yeah, sorry.
I'm sure it does something. Yeah.
What's the ABL part stand for?
You know, Alison kinase something or other.
It's a name of a person, no?
Yeah, it's kinase. So we call it the able kinase.
Yeah.
Yeah.
Yeah, it's a name of a person, probably the person identified this kind of.
Yep.
So this molecule has now been a lifesaver, and there's many patients that live with this disease, and it's like a chronic disease, so they have to take this medication for the rest of their life, but it's like the disease can be controlled. However, there's a lot of patients, so before I get there, because this is very specific for ABL, so this molecule was designed to bind the binding pocket, but because the human genome has more than 500 kinases, as you can imagine, it had to be designed in a very specific way that only targets this kinase. Otherwise, it's going to inhibit many other kinases that shouldn't inhibit, because we want to make sure all the other kinases are working properly. So they had to design it in a way that is very, very specific for the pocket of this kinase. So as a consequence, it's very easy to acquire resistance to this drug. So what this means is that cells find ways to mutate this kinase in these ATP binding pockets, and just a single mutation, and this is a very common one, so this threonine that is replaced by isoleucine, but there's many others. This is just the most common one, that by having this mutation makes the imatinib unable to bind to the ATP binding pocket, and so basically the patients become resistant to this drug. So they're not going to respond to the drug anymore, so this is going to relapse, and so alternatives have to be found. So there's now second and third generation drugs, and most of them, or all of them, are still using the same strategies. Yes?
How long are they actually looking at before the patients become resistant?
So it has been seen like around about 5 to 8 years, so patients can
That's
become,
pretty good.
yeah, it's pretty good, but still
Yeah.
it's a resistance. So some patients develop it much earlier, but it has definitely increased the overall survival of these patients, and some of them do acquire just after a few years, after some years of
chronic as well too. I suppose, looking at sort of more an underlying issue there.
Apparently, there's a lot of side effects as well, like some rashes in the skin, because it's impossible not to have side effects when it's a kinase, when it's an ATP binding pocket.
Oh, really? Okay.
There's still some side effects.
Another
Which
one, Jordan.
yes,
Sorry to interrupt. I wanted to ask, so the BCR-ABL, it's in the chronic myeloid leukemia. Are there any other cancers that it's been identified in?
I don't think so. This is
So it's like
some other leukemias they might have it, but this.
it's specific to leukemia.
So the unique thing about chronic myeloid leukemia is that so many cancers, so all the other cancers have several mutations, and it's hard to just pinpoint one mutation.
Yeah.
For chronic myeloid leukemia patients, 95% of them have these mutations.
Wow.
So
okay,
then it's
right.
that's why it was considered a model cancer, because it's a very unique characteristic mutation, and then it was easy to target in the sense that it's just this mutation.
Thank you.
And yes, so all these new generations of drugs, they all have similar mode of action, so they are still mimetics of ATP to insert in the binding pocket. So resistance still occurs, and so this means that we need to think about strategies and new types of drugs to target different cancers. So this is to say that this is just an example, but this is something that occurs with many cancers. So there's a lot of kinase inhibitors, in fact, that are used to treat cancer, and examples are in the melanoma space, and they all end up having similar issues. So drug resistance can evolve to these small molecule drugs because they are small. It's easy for the target to evolve in a way that a single mutation is going to render the molecule inactive. Yes.
Sorry, how are you identifying the drug resistance? Is it just like frequent testing, or is it like symptoms coming back? Or
yeah, so I guess symptoms coming back, and then just you can do a genetic test and making sure it's what's happening. Yeah.
My question was quite similar to the one you just said, but how frequently is drug resistance sort of observed?
Very frequently. So small molecule drugs, like even for chemotherapy, so I'm sure you will have heard that people that have their cancers coming back often relates to becoming resistant to that treatment. And there's several strategies for that cancer cells have evolved to develop resistance to drugs. Some of them overexpress these membrane channels that efflux the drug outside, so they just pump the drug out. And the problem with drug resistance is once a cancer is resistant to one drug, sometimes they are multi-drug resistant. So they become resistant to many other drugs. Because some of the mechanisms, they just work for any drug, so the cell just evolve in a way that they can pump out all these drugs out. Other types of acquired drug resistance is changing the location, the hotspot where the drug binds to, like the case of this example shown here, but there's many other strategies. Okay? Other times they just inactivate one path and activate other to make the cells proliferative. Yes.
And so when you incur drug resistance, do you often see random excretions of electrolytes? Because I know that oncology patients, when they go and get blood tests, they always get electrolyte liver function tests and they get a full blood count. And that's mostly what they get. So are they able to see it through those tests?
I think it relates to the acidosis around the tumor, because if the tumor is very acidic, it means, I will talk a little bit about that towards the end of my talk, so hopefully that will answer some of those questions. All right, so talking about drug resistance in cancer, so this is really a very brushed overview on how drug resistance can occur. But when we have a tumor, so tumor is very heterogeneous. So drugs are developed to target some types of mutations in the cell, but then there's cells that might have other types of mutation, and there's also what is referred to as a dormant cell. So drugs are normally developed to target these highly proliferative cells, and there's always a population of cells that is referred to as dormant or non-proliferative. So very few drugs can really kill these normally drugs are they can't kill these non-proliferative cells. So which means that, so the other problem is that these dormant cells can also evade immune detection. So they're just kind of slow cycling, they don't replicate, they are just pretending they are not there, but they are there. And so these cells can evade immune detection, which make them not susceptible to immunotherapy. So immunotherapy, they might be very successful in activating the immune system to kill the proliferative cells that express the right receptors on their surface. But then these slow cycling cells or the non-proliferative cells, they're not going to express those receptors on their surface. And chemotherapy and target therapies, they are normally developed to modulate proteins or DNA in these proliferative cells. So which means they're not going to be active again in these dormant cells. So dormant cells are really, really difficult to kill. And as a consequence, when we treat these heterogeneous tumors with the current therapies, what's happening, there's always a population of cells that is going to survive the treatment. So these cells that survive to treatment, if you take them off drug, then eventually they can become proliferative. That's what we call drug holiday. But if you keep treating them with drug, this population is there, dormant, until it gains, so it normally can be phenotypically similar to the proliferative cells. But as they are being treated with drugs, so they are being challenged with drug, they sometimes accumulate genetic mutations that then make them proliferative again. And then these mutations make them insensitive to the current drugs. So as a consequence, these dormant cells survive, and then they survive drug treatment, then they develop resistance mechanisms, and they regain the ability to proliferate, and they form new drug-resistant tumors. And these are the ones that then are very difficult to treat because they are resistant to current therapies. Yes?
Is there any markers or some more screening methods for these dormant cancers?
It's very difficult to detect them. Yes, there's some markers because they are slow cycling, so there's ways to identify that they are not there. But most of the markers that exist are for proliferative cells, so that's the challenge. So even for diagnostics, it's very difficult to identify where they are because they are kind of pretending they're not dead. They are pretending they are dead, but they're not. So those cells then they form new drug-resistant tumors, and they might form metastases in other organs. And then eventually, when they gain ability to proliferate again, they can develop new tumors. So there's a need for therapies that can kill these non-proliferative cells, and this is kind of a big challenge. So we really need to look into new targets, maybe targeting the drug-resistant pathways, and also to new therapeutic modalities that are not so relying on small molecule drugs that can easily induce acquired resistance. So the main problem that we are trying to work on is how can we find and kill all these tumor cells, not just the highly proliferative cells, but also the dormant and drug-resistant cells that are difficult to kill with current therapies. And just giving a very broad overview on different modalities or drug modalities that exist. So we have already talked about small molecule drugs. These ones that we can take orally and can cross the membrane. So these are cell permeable. Yes.
At what point does a drug become impermeable to a cell membrane?
They become in small molecule drugs. So small molecule drugs, they can still cross membranes, but if there's these efflux pumps that pump out the drug, then they go in, but then they get out as well. Other drugs are impermeable to the drug, to the cell, like the antibodies are impermeable to cells, so they can't really cross the cell membrane because they are too big to cross cell membranes.
Okay.
We'll be talking a little bit more about that. So small molecule drugs, they are cell permeable. They can modulate intercellular targets, so they are normally used when we want to modulate intercellular targets. But they are not cell-selective in the sense that they can enter all types of tissues, which means they can induce off-target effects and toxicity. They can only be used to target this type of proteins that have small pockets, like the kinase I've just mentioned to you, and they can easily induce acquired resistance. So small molecule drugs, they are great, but they do have their limitations as well. Biologicals, they have been very useful to treat some cancers, and this includes also the immunotherapy where antibodies are developed to induce the immune system. They are very specific, so they are really good in the sense that it's more difficult to develop resistance against them, but they can only be used to target cell surface or extracellular receptors because they cannot cross the cell membrane. So they're not going to be useful. So there's a lot of cancer targets that could be targeted if we could get those antibodies or biologicals inside the cell, which at the moment we can't. Yes.
I was wondering what's different in the the dormant cells that means that it can't be targeted by chemo or with targeted therapies. Like
What's different? So with chemotherapy, you are targeting cells that are rapidly dividing. So they target the DNA on cells that are rapidly dividing. So they're going to target healthy cells that also are like the hair follicles and our digestive tract. That's why there's so many side effects with chemotherapy. But they still kill tumor cells better than they kill healthy cells. So there's still a lot of benefits in using chemotherapy for some types of cancers. So as I was saying, biologicals, they can be specific. They can only target cell surface receptors. But the problem is that they're not gonna target cell subpopulations that don't have the antigen, that don't have that receptor expressed on the surface. And there's many patients for which the biologics are not gonna work because they don't have the antigen, so they don't have the membrane receptor that is required for the biologicals to work. So many patients are intrinsically resistant, so it's not gonna work for all the patients. So there's now new modalities that are being used to kind of add to these two major traditional types of therapies. And one of them is antibody-drug conjugates. So this type of modality, what it does is, as the name suggests, is putting a small molecule together with an antibody. What they're trying to do with these is to make the drug go straight into the tumor cells. So it's more targeted because small molecule drugs go to any cell, so in this way, just make it more targeted. So the antibody is designed to specifically recognize a membrane receptor on the tumor cells, but then the drug is what's going to do the job. But in this case, this is dependent on the antibody getting inside the cell, and it does that via endocytosis. And there's problems with that, which we're going to be talking about just next. So we still have the same problems as with biologicals or with small molecule drugs that can occur with these. But it's just new strategies that can help and have benefits in some patients. And the new other strategy that is you might have heard about, mRNA vaccines for cancer. So this is something that is is kind of the big thing that is now being explored. So these strategies, in these strategies, the mRNA, it has to be stabilized and delivered using what is called the lipid nanoparticles. So these lipid nanoparticles are basically liposomes, where that's kind of protect the mRNA, because mRNA is very unstable, and helps the delivery of these mRNA. The mRNA is also very polar, it's very negatively charged. It's very difficult to get these polar molecules across cell membranes. So with these lipid nanoparticles, then it helps stabilize the mRNA and make them get inside the cell. The problem is there's a lot of science and a lot of people that is just focusing in improving these lipid nanoparticle structures. Yes?
Is the LNP sort of similar to like extracellular vesicle delivery system?
In a way, yes, because it's basically a lipid bilayer. So it's a vesicle made of a lipid bilayer, and that can carry mRNA like extra vesicles. So they are just carrying. But these ones are obviously made in the lab, and they are very controlled. So they use synthetic lipids. They know exactly the lipid composition, and then they modulate and they use synthetic lipids. So some of the lipids they use are what is called ionizable lipids. So this is a lipid that becomes charged when it's in an acidic pH. So this is to help them the delivery inside the cell when it's inside the endosome. I will go and touch that next. So the problem with these strategies is there's still a lot of endosomal entropy. Even with all these strategies to improve the delivery of these lipid nanoparticles, many still are retained inside the endosomes. And these type of strategies also have a tendency to then, when you look into the biodistribution, many of them end up in the liver, which means it's suboptimal. Instead of going to the right organ, you have a lot of these being loaded into the liver, which can induce hepatic toxicity. So this is just something to keep in mind. So the challenge we have here, yes.
Sorry, just in terms of the antibody drug conjugates, let's say, because you said that it was kind of both a small molecule drug and the biologicals. Let's say if someone goes into the antibody drug, can they like kind of go back to the biologicals, or do they have to stay in that drug?
So these are going to be different molecules at the end. It really depends on the cancer. So there's not a really a workflow or a regimen that you're going to take for it. It really depends on the type of cancer. And these are just new strategies that are appearing or that they are being developed as strategies to treat some cancer cancers. Yeah. So the challenge that exists at the moment is trying to combine cell selectivity to reduce toxicity, have good intercellular delivery, something that can get inside the cell, and still have this high binding affinity for the target, so for the protein that we are trying to modulate and inhibit inside the cell that can induce the cancer. So before I get into a little bit more detail on peptides and why we are working on peptides, I just wanted to give you just tell you about the Lipinski rule of 5. So this is a rule. This is a rules that have been created by this researcher that kind of gives you the drug likeness of a compound. So there was this set of rules where molecules had to have these properties. So they shouldn't be more than have more than 5 hydrogen bond donors. They should not have more than 10 hydrogen bond receptors. They should not be bigger than 500 Daltons, and they should have good partition into membrane. So this is basically a set of chemical properties that a molecule should have to be something that can get inside the cell and target an intercellular target. So this set of rules has been kind of controlling the drug development programs for finding new drugs. So basically, if a drug falls out of this rule of 5, it's not even considered for the screening programs. So when we are looking for new molecules to inhibit a target, what people do, they use libraries of compounds that follow this set of rules. So which means everything that is fall out of this is not used for these drug screening programs. So what these drug screening programs are looking for is molecules that can be orally active. So it means you can take it through your, can take them orally, and they can go through your digestive system and getting into the systemic system. So they're going to go into circulation. Yes.
How big are the libraries that they use for
It
these?
can be thousands of compounds. So there's whenever anyone is trying to find a new molecule to hit the target, they start with a complex library of different molecules that all fall into these rules. And then when they find some hits, then they look into that hit and they try to further modify it. Then they kind of improve that hit. And then they do some, they call it medicinal chemistry changes to improve that molecule and the affinity of that molecule for the target.
The library I screened had 6 million.
Can be,
There are libraries that are much, much bigger. It depends what you're looking for.
yes, but they try to have like a complex library that has different properties. Okay, but normally they are fixed on these molecules that follow these rules, the rule of 5. So what those rules mean is that then we have a set of molecules that what they do, I call them the low 5 molecules because they can't have more than any of these 5 things. They can't be more than 500 Daltons, etc. So I call them the low 5 molecules. So what these low 5 molecules do is that they can just cross membranes and fat layers, okay? So for this set of molecules, cell permeability, membrane permeability, and ability to reach an intercell target is the same. So you can get through your can take orally, you can cross your intestine, go to to circulation, go to your blood, and then can be delivered to the different organs, and eventually it's going to get into the cell, to the tumor cell, and hit the intercell target. But in this way, you're going to get cells in this drug into every cell of your body because it just goes through any type of membrane. Okay, so I call these, so basically the low 5 molecules, they can cross fat layers. Okay, and they are not specific to any type of cell. Yes.
So say a molecule violates just one of the rules, is it then like automatically not a viable treatment strategy?
Normally they just don't consider, so they just exclude those.
Okay.
But they might have a scaffold that follows all the rules, and then they change slightly, might violate one of the rules, but it's still considered.
Okay.
So obviously there's a balance between that. But yeah, they started with molecules or with scaffolds of molecules that kind of follow those rules. All right, so the thing we have been working on, I call it the solution 3-in-1. We're trying to use peptides that can target tumor cells, that can deliver drugs in the cytosol, and that can modulate intercell targets. So peptides, if you remember from your biochemistry, these are kind of mini proteins. They fall in size between small molecule drugs and the biologicals. Okay? So biologicals normally are larger than 5 kilodaltons. Small molecules are below 500 daltons. So peptides are in between 500 daltons and 5 kilodaltons. So it's normally molecules that have between 2 and 50 amino acids. So the reason why we are interested in peptides is because they have larger surface area than small molecule drugs, which means they should be able to inhibit or modulate proteins or areas in the protein that are larger than what small molecule drugs can do. So in theory, we can achieve higher specificity and we can target protein-protein interactions. So normally small molecule drugs can only target pockets in enzymes, whereas with peptides, we're going to be able to inhibit protein-protein interactions that are important and involved in cancer pathways. So it means we inhibit these interactions, and then the pathway is no longer continuous. And the other advantage is because peptides are larger, we have larger flexibility to design if resistance occurs. Okay, if there's a mutation on the target, then we can redesign the peptide just to mutate also one amino acid, and in theory we should be able to regain affinity for the target. These are the advantages of peptides. The problem is, interesting peptides, they do not follow the rule of 5. So they have a larger surface area. We know that they can achieve higher specificity for the target. They have all these properties. But if we really want to use peptides that have higher surface area, they're gonna be longer than 5 amino acids. So they're gonna be breaking all the rule of 5 rules. And the other thing is peptides, they do have a lot of polar regions, so they have a lot of hydrogen bond donors and receptors. They can also have charges. So which means that they're not going to interact with cell membrane just by going through the cell membrane diffusion. So they're going to have polar and polar interactions with cell membranes. So for peptides, cell permeability is not the same as membrane permeability and cell penetration. These are different things. So that's the so what we're trying to do is, because these are different things, so it's not something that can just go through all the fat layers, all the cell membranes. We want to use this challenge as an opportunity to develop molecules that now are specific for certain membranes. Before we go into that, just wanted to give you a brief overview on how can drugs enter inside cells. So the first is via passive diffusion, which is what small molecule drugs, they just go through the lipid membrane. So this occurs with small molecule drugs and also some peptides that are lipophilic, so they just go through the membrane by passive diffusion. There are some molecules that can go through via an active transport, so they bind to a transport on the membrane, and then it can pump the molecule inside the cell. There are molecules, but then there's peptides that can do what we refer to as direct translocation. So this is a type of mechanism that involves peptide-lipid interactions that are not just going through fat layers. So it's involving polar and apolar interactions. And this is what amphipathic peptides normally do. And then all the other things normally go through endocytosis. There's many endocytic pathways, so it can be, some of them is specific endocytic pathways, others just use any of those endocytic pathways. And this is what large proteins do, like the antibody drug conjugates. So they just go through, enter inside the cell through endocytosis. And this is also what the mRNA lipid nanoparticles do. So when we are delivering mRNA vaccines, they basically fuse, the lipid nanoparticles fuse with the cell membrane, and then it just gets into the cell through this endocytic pathway. So what's happened is that we often have endosomal entrapment, which means the drug of interest is inside these endosomes, and only a small percentage can actually escape the endosomes. So the endosomal escape is can be promoted via peptide-lipid interactions or through membrane fusion. So that's what happens with the lipid nanoparticles. So remember I said they had these chargeable lipids. So the endosomes have an acidic pH, so it's more acidic than the rest of the environments. So when they get into this more acidic pH, those lipids become positively charged, which then helps fusing with the membrane, which is negatively charged. And the membranes of the endosomes are also more negatively charged. So these play with the charges, helps this membrane fusion, and then it's going to release the mRNA and other molecules inside the cell. Okay? But this is a strategy that really is not very efficient, and there's a large amount of drug that, so actually we need to inject a lot more drug than we would need if we had an efficient delivery. All right, so this is the problem that we have when we want to target intercellular or molecular cell proteins. But in my group, we have also been trying to see what other strategies can we use. So new therapeutic targets for cancer. So these hallmarks of cancer, many of them are mediated by mechanisms that involve lipids, and they can promote these hallmarks and enable characteristics of cancer. And one of the things that happens is remodeling of lipid membranes. This occurs during cancer progression. So what we have been trying to look at is, can we use cell membranes as a target to help peptides specifically go through cancer cell membranes? So if you remember from biochemistry, the cell membranes are made of a lipid bilayer that contain different types of lipids, like glycerophospholipids, phenolmyelin, and cholesterol. And within the glycerophospholipids, they can have different head groups, here represented by these colors. They obviously have different chemistries, which confer to them different chemical properties. And they also confer to them different roles in the cell membrane. So what I wanted to emphasize here is that cell membranes are chemically diverse, and they are also asymmetric, which means that the extracellular part, so the outer layer that is exposed to the outside, is different from the inner layer. And so one of the key features of healthy cells is that we don't have negatively charged lipids on the outer layer. The negatively charged lipids, here shown with this red lipid, they are only located in the inner leaflet. So we don't have negatively charged lipids outside. And the other feature that is normally observed in healthy cells is that we have cholesterol and sphingomyelin in the outer leaflet and not in the inner leaflet. So, which means that we have more rigidity in the outer parts than the inner part of the membrane. So this asymmetry is lost when we go to cancer cells. So in cancer cells, these lipids are all scrambled, okay? This is maintained by enzymes. When you get to cancer cells, because we are just rapidly proliferating, this is not maintained. So the symmetry is lost. The surface of cancer cells is more negatively charged, and because now negatively charged phospholipids are surface exposed, and there's also transmembrane potential, which is more negative in cancer cells. So all of these make the surface of cancer cells very different from the surface of healthy cells. So we have been looking into membranes as target to modulate selectivity to tumor cells. And we have been doing that with peptides, which are part of a host defense mechanism of organisms. So these peptides, also referred to sometimes as antimicrobial peptides, they are often positively charged, and they are expressed in any organism. Okay? All the organisms express this type of peptides as a way to fight bacterial infections normally. The examples I'm going to talk to you about today are gomazine, so that's one of the peptides I've been working on, which is expressed in this Brazilian spider, and it's expressed in the hemocytes. So hemocytes are kind of the white blood cells they don't have blood, they have hemolymph of these animals. Okay, so it's expressed as part of the immune system. This peptide contains 18 amino acids, has 2 disulfide bonds, and forms a structure that is often referred as a beta hairpin. So it looks like a hairpin, but it has a an antiparallel beta sheet that is stabilized by 2 disulfide bonds, which are shown here in yellow. So that's the sequence of the peptide here. So that's the disulfide bonds, just showing that it's quite constrained structure. This peptide was shown to have selective toxicity against melanoma cells. This was quite interesting. And then what we have done was try to make this peptide even more stable by linking the 2 amines. So we stabilize the peptide, and then we look into others that were similar, like tachyplasmin-1, and they all have very similar properties. Tachyplasmin-1 is also positively charged host defense peptide, and both these peptides have selectivity for melanoma cells. So it means that they can kill melanoma cells with higher efficiency than any other type of cell. In some cases, we saw 40-fold selectivity, and we can modulate these peptides even further to make them even more selective for cancer cells, or melanoma cells in this case. We found that they kill those cells because they bind to negatively charged membranes. So we first did that with model membranes, but we have also confirmed with cells. So these peptides do not bind to cells or membranes that have their membrane neutral, but they bind to cancer cells that are negatively charged. And they can disrupt those membranes, neutralize those cells, and eventually kill the cells via membrane colonization. So we wanted to understand why were they selected for melanoma cells, confirming it was related to the charge of those cells. We have done lipidomics, and we looked into the composition of the cell membrane of a panel of cells. And what we have found was that in melanoma cells, so these ones shown here on the top, they have lower amounts of sphingomyelin and cholesterol. So which means that they are expected to be more fluid than the non-cancer cells, and in fact, more fluid than many of other types of cancer. But what we also found was that melanoma cells were indeed more negatively charged than any other types of cells we have tested. So the melanoma ones are here on the top, and as you can see, they are very negatively charged. So we could confirm that the cell surface of melanoma cells is more negative, and that's why those peptides target those cells. Then we wanted to understand, can we use these peptides as a strategy to kill drug-resistant cancer cells? So what we have done was create drug-resistant models by treating melanoma cells with dabrafenib, which is a standard of care for patients that carry this a mutation, which is called the BRAF V600 mutation. So this is a kinase that is also constitutively active in many melanoma patients, and this drug inhibits this kinase. And what we have found was, as we keep incubating the cells with this drug, at the beginning we have these drug-tolerant persistence that survive the treatment, and eventually the cells regain ability to proliferate and become resistant to the drug, and they proliferate at a rate that is similar to the original untreated cells. When we treat our peptides, when we treat those cells with the peptides, we found that the peptide could kill the dormant cells, but could also kill the drug-resistant cells. So this is the cyclic tachyplasm and the cyclic gomosine. Yes?
In that diagram, what does drug holiday?
So drug holiday means really drug holiday. So it means you can take patients out of the drug to see if these dormant cells that so you kill all the proliferative cells, there's the dormant cells that survive. If you take them off drug quickly, eventually these dormant cells can regain ability to proliferate, and then they can be killed again by the drug. But if you keep treating those cells with drug, eventually they become genetically different, and then they gain mutations, and then they become proliferative, and you can't kill them with drug. So in our case, we found. Yes.
Okay. Have these been used on like actual real patients yet, or it's just been in the lab?
No, we're just testing them in the lab. But this is dabrafenib is a drug that's used to treat patients, and this actually occurs. So patients gain resistance to this drug within 6 months. It's very common for patients that are on this drug to gain resistance to the drug. We made it in the lab in 3 months normally. And this is actually a similar time frame that happens in patients as well.
You guys are going to have to let it finish.
Yes.
Because we've got like 2 minutes left.
All right, I'll go very quickly now.
Okay.
Let's save those questions so I can finish here. So those peptides, they can kill the drug proliferative cells, the dormant cells, and also the drug-resistant cells, which was really exciting. But then we wanted to see, can the cells develop resistance towards the peptides? And what we have found was we treated the cells in the same way as we have treated with the small molecule drug, with dabrafenib, for more than 3 months. And we found that the cells could not gain resistance to those peptides. So they could not find mechanisms to fight the activity of those peptides. So this was really exciting. They could not adapt treatment, the peptide treatment. We're looking, because we know they target the cell membrane, we wanted to see if there was changes in the lipidome while we're treating the cells with the drug, with peptide, and the cells did not change their lipid composition. And we also confirmed that using proteomics, so we just look into variation of expression of proteins that are important for the synthesis of structural lipids that make up the cell membrane. And when they are treated with peptide, there was not really much change in the expression of those proteins that are involved in lipid metabolism. So basically, the lipids that make up the cell membrane cannot be modified, even if they are doing treatment with and other peptides. So now what we are trying to do is use those peptides as a strategy to now deliver drugs inside cancer cells. I'm just going to go very briefly fast here. So these peptides can enter inside cancer cells. So in addition to be able to kill and disrupt their membrane, they can enter inside cells at lower concentrations than their membrane disruptive properties. And so we can use them as a scaffold to stabilize sequences that could be used to inhibit proteins of interest inside the cell. So that's what we have done with our peptides. We have developed them to inhibit LDH5. LDH5 is an enzyme that is overexpressed in many tumors. Tumor cells, they normally reprogram their metabolism for production of ATP, and they use what is referred to as aerobic glycolysis or Warburg effect. So it just cells instead of using the cancer cells instead of using oxidative phosphorylation to produce ATP, they keep using glycolysis as a strategy to produce ATP at a faster rate. So although glycolysis is not as efficient, it's faster production. So as a consequence, so there's this LDH5 that helps regenerating NAD+, that's going to be reused on the glycolysis and converts pyruvate into lactate. This lactate is expressed and is excreted into the tumor environment, and that's what creates this acidosis, so a more positively charged environment around the tumor. Right? So there's more protons in the tumor because this is an acidic molecule. These changes helps tumor cells evade so they help with the immune suppression. So these cells that overexpress this LDH5, they are more resistant to treatment. They are more resistant to treatment, and they are more difficult to treat as well with immunotherapy. So the increase on the production of lactate facilitates drug resistance. So we thought this would be a good target to try to improve the overall survival of patients, because if we inhibit this LDH5, we are also inhibiting all the strategies that tumor cells have to improve drug resistance. So LDH5 has been an enzyme that has been tried to be targeted by pharma companies. This is an enzyme that is made of, as a quaternary structure, is made of 4 monomers that tetramerize to form this active structure. Pharma company has been looking to target the active sites, and so they find mimetics of NADH or pyruvate that can fit into the active sites. So they are competitive inhibitors. So they bind to the active sites, try to outcompete the substrate. The problem with all these molecules is that they all failed because they were not really good. They didn't have good permeability, and the other problem was because they are NADH mimetics. There's many oxidorhydrotases that also have off-target effects. So the strategy I've been trying to do is to inhibit the dimer-dimer interface. So we inhibit this tetrameric formation to help. So because if the enzyme is not in a tetrameric shape, it's not going to be active. So what we're trying to do is to disassemble these different the 4 units, so then the enzyme is no longer active. So we have done that using mimetics of this dimerization interface, which has a beta-sheet interaction. So we have synthesized peptides that mimic this interface, and then we have grafted into our cyclic gomazine and use it as a scaffold. So basically, then we have done a lot of iterations of this grafting. We have done some rational improvements and in silico library screening to find then a molecule that now we know can cross the cell membrane, enter inside the cell, and inhibit these protein-protein interactions that are involved in the activity of LDH5. So in this way, we could inhibit the LDH5 activity and inhibit proliferation of these cancer cells. So this is still work in progress. But just to summarize, and so you all can go, we started with an antimicrobial peptide. We have cyclized to be sure they can cross the membrane, and we are now using it as a scaffold to deliver active sequences that can inhibit proteins that are involved in cancer progression. All right, I'll leave it here because we are really on time. And just I want to thank my team. So this is my research team at TRI. Elliot can tell you about them because he spent some time in our lab last year. And yeah, and I want to say that we have a very diverse team, and we are very proud because we know that diverse teams are better than me.
Let's clap.
Thank you.
Yeah. I'm